Welcome everyone. Today we are going to learn about sphingolipids in more details. In my previous video I have mentioned that there are three major categories of lipids present in the plasma membrane. The first is phosphoglycerides, second is sphingolipids, and third is cholesterol. In my previous video I have mentioned or discussed about phosphoglycerides in more details. So today we are going to learn about sphingolipids in more details. So today's topic is sphingolipids. Sphingolipids. At first, we will learn the components of sphingolipids. There are three components present in the sphingolipids. The first is a molecule called sphingosine. From this molecule name, the sphingolipids gets the name sphingolipids. So sphingosine is the basis. Now, in my previous video, phosphoglycerides, I have mentioned that glycerol is the backbone of phosphoglycerides. Now, here sphingosine is the backbone. This is the backbone. Okay, remember this. Second component of sphingolipids is one fatty acid chain are actually present. So one fatty acid chain actually attached with sphingosine. And third component is head group. Just like phosphoglycerides has alcohol head groups, it also has head groups. So it also has head groups. Now let's talk about each component in more details. Sphingosine. At first we will talk about sphingosine. Sphingosine molecule is actually derived from an amino acid called serine. So from serine amino acids, sphingosine is actually made. Basically what happens in the endoplasmic reticulum, serine molecule is actually attached with one fatty acid. So in the serine there is first carbon, second carbon, third carbon, fourth carbon. Now fourth carbon is actually double bonds with fifth carbon. With the fifth carbon, one fatty acid is actually attached. Okay. And this structure, actually fatty acid attached in the endoplasmic reticulum. So this structure, serine attached with one fatty acid, is called sphingosine. So this is the sphingosine. Okay. So basically sphingosine is made up of serine. And now serine with the fatty acids is sphingosine, and this is the backbone of sphingolipids. I think you have understood. Now, in the endoplasmic reticulum, another fatty acid that I have mentioned, one fatty acid chain attached with the second carbon of the serine. Then when this another fatty acid is actually attached with the sphingosine, then it becomes, so I can write this structure is sphingosine. Sphingosine. This is the first structure. Then when another fatty acid is attached, this structure is called ceramide. Ceramide. So this structure togetherly we will call ceramide. And when ceramide is made, from this ceramide molecules actually transport out from endoplasmic reticulum to Golgi complex. Golgi complex. And in the Golgi complex there are some enzymes present that attaches head group with the first carbon. So head group is attached with the first carbon of serine, and ultimately in the this reaction occurs in the Golgi complex, and ultimately we get with the third step is sphingolipids. So this is the structure of sphingolipids. So I have discussed about sphingosine. Now I want to mention some facts about fatty acids. So let's talk about fatty acids in the sphingolipids. In my previous video, phosphoglycerides, I have mentioned that the phosphoglycerides also contains fatty acids, two fatty acids. Just like these two fatty acids, phosphoglycerides also contain two fatty acids. Now the difference is here, in case of phosphoglycerides, okay, the fatty acid chain generally it contains carbon number 16 or carbon number 18 fatty acids. So the fatty acids contain carbon number 16 or carbon number 18. Generally it has been found. But also C14 and C20 fatty acids are also found. That means those fatty acids contain carbon number 20 or 14. 14 number of carbon that contains the fatty acids called myristic acid. So those fatty acids are C14, we call myristic acid. Myristic acid. And in case of C20, we call it arachidonic acid. So in case of phosphoglycerides, you see the carbon number of the fatty acid is 16 or 18, in some cases 14 and 20. But in case of sphingolipids, the length of the, this is for sphingolipids, the length of the fatty acids is generally longer, and also another information you remember, more saturated than phosphoglycerides. Remember this. This is very important information. Why I am telling so? Let's see, the length of the fatty acids in case of sphingolipids is longer and more saturated. At first, I want to mention that the length of the fatty acids, the number of carbon here can up to carbon C26. So the number of carbon can goes up to C26. So it is most of the cases longer than the fatty acids present in the phosphoglycerides. Also, it is more saturated. Now, what is saturated? I have also discussed in my phosphoglycerides video that fatty acids can have C double bond C, mean double carbon-carbon double bonds. A fatty acid can contain C double bonds. Those fatty acids contain C double bonds we call unsaturated fatty acid. Unsaturated fatty acid, and those doesn't contain C double bonds we call saturated fatty acids. Saturated. Now the difference is here, a fatty acid that does not contain a C double bonds is actually straight. Is straight in structure. But if the fatty acids, let's suppose here contains a C double bond, then there will be a kink. What I am talking about? A kink or bend in the structure. Where there is a C double bond, there will be a kink or bend in the structure. And this has a very important role in the fluidity of the plasma membrane. As you can see, the presence of carbon-carbon double bonds has an important role in the structure of the fatty acids, because where there is a carbon-carbon double bond, there will be a kink or bend. Let's suppose if there is any carbon-carbon double bonds is present here, carbon-carbon double bond is present here, the structure will not be like this, it will like this. So where there is a CC double bond, there will be a kink. Okay, so it will actually bend the structure of the fatty acids, and also it has an important role, these unsaturated fatty acids have important role in the fluidity of the plasma membrane, that I will talk separately on in different videos. So as these sphingolipids contain longer and more saturated, that means the fatty acids are straight, so this is saturated fatty acid, saturated, and these are unsaturated fatty acids. Example of unsaturated fatty acids. So this is saturated and this is unsaturated. So this is one information you can remember, and that's why the sphingolipids is more stable than phosphoglycerides. So up to we have learned about the fatty acid chains of the sphingolipids. Now let's talk about head groups of the sphingolipids. Now the head group of the sphingolipids, could be phosphocholine. Remember, if you have watched my previous video, I have mentioned, choline, the spelling will be CHOLINE. In my previous video, phosphoglycerides, I have mentioned that the head group of phosphoglycerides can be phosphocholine. So in case of sphingolipids also, phosphocholine can be a head group, or phosphoethanolamine. Phosphoethanolamine can be the head group. This phosphoethanolamine can also be the head group of phosphoglycerides. So here basically the two head groups actually similar in case of both phosphoglycerides and sphingolipids. Now those sphingolipids. Remember, this is the structure of sphingolipids, and those sphingolipids contain this type of head groups are called sphingomyelin. Remember the term sphingomyelin. The basic structure will be same, serine, two fatty acid chains attached, but the head group just is phosphocholine or phosphoethylamine. We call it sphingomyelin. Now the name suggests here myelin, that means it is present in the nervous tissue of our body. As basically in the myelin sheath, the sphingomyelin lipid molecules are abundantly present. Sphingomyelin is the most abundant sphingolipids in the plasma membrane. Like in case of phosphoglycerides, the phosphatidylcholine is the most abundant phosphoglycerides in the plasma membrane. So this phosphocholine is also in case of phosphoglycerides abundant, and sphingomyelin is also abundant sphingolipids in the plasma membrane. So this is one kind of sphingolipid. Now the head group could be a sugar molecule, and the sugar could be monosaccharide in nature. Monosaccharide. Example like glucose or galactose. So see here, the head group could be a monosaccharide like glucose or galactose. Then we call this type of sphingolipids cerebroside. Cerebroside. Okay. Now here also a term comes that is related with our nervous system or nervous tissue, that is cerebro. Now if the head group is glucose, we call this sphingolipids glucocerebroside. Glucocerebroside. If the head group is galactose, we call the sphingolipids galactocerebroside. See, this is the naming of the sphingolipids. Now one information I want to share, that this galactocerebroside is abundantly found in the myelin sheath, and mice that lack the enzyme, that can attach the galactose to the head group, show or exhibit some symptoms, neurological symptoms like severe muscular tremors, and eventually paralysis. Okay, so this is one information you can remember for galactocerebroside. Now the sugar molecules could be oligosaccharides. This is another example, oligosaccharide. If the sugar molecule is oligosaccharide, then we call this type of sphingolipids gangliosides. Examples of gangliosides are GM1, GM2, GM3, etc. So what we have learned here, that if the head group is phosphocholine or phosphoethylamine, we call it sphingomyelin. This is one kind of sphingolipids. If the head group is monosaccharide, we call it cerebroside. This is another type of sphingolipids. If the head group is oligosaccharide, we call it gangliosides. This is another type of sphingolipids. Now in this case also I want to mention one information, that humans, those lack GM3 sphingolipids, they also show some neurological disorders, characteristics characterized by severe seizures and blindness. In human that lack GM3, they show severe seizures and blindness. Okay, remember this for GM3. Now one information I want to mention here for the oligosaccharide. As I have mentioned, this is oligosaccharide, means many sugar molecules are present, not like the glucose or galactose, but many different sugar molecules can be present. But In case of oligosaccharides, one sugar is always there. Is an example of sugar is called sialic acid. Remember the name, sialic acid. This acid is actually carries negative charge. They carry negative charge. This sugar, sialic acid, yes, this sugar must be present in the oligosaccharides, and it has been found that all the oligosaccharides present in the gangliosides contain sialic acids. Now in case of human, there are various sialic acids are present, but one of the sialic acids is a common, it has been found. The name of the sialic acids I'm writing here. The name of the sialic acids, okay, I'm writing here the name of the sialic acids is N-acetyl neurominic acid. In short, we can say N-acetyl neurominic acid. It is the most common sialic acid present in the humans as oligosaccharide or as a sugar in the oligosaccharides. Okay, now another information I want to share. Okay, what I am writing here. So one information you can also remember for gangliosides, that in case of gangliosides, cholera toxin gets entry into a cell with binding with one ganglioside called GM1. So cholera toxin is an exotoxin. It binds with GM1 present in the plasma membrane, then it can enter inside the cell. Another example of toxin is called botulinum toxin. Botulinum toxin. It can also enter inside the cells through the help of these gangliosides. Another is influenza virus, and also polyomavirus. They can get entry inside the cells with the help of these gangliosides. So you can summarize all the informations here. Sphingolipids. So in this video I have actually discussed about sphingolipids. In my next video I will talk about in more details on about cholesterol. So hope this video will help you. Thank you for watching and always keep learning.